will talk about data that I collected in the lab of John Reynolds when I was a postdoc at the Salk Institute. In the meantime, I've moved to Washington University, where we study visual segmentation in my lab. So I'll talk about borders in visual scenes. As many of you know, borders are an important component of everyday visual scenes and artificial visual scenes, including this interesting example here on the left. Borders occur whenever an object meets background or whenever objects occlude each other. So what does a visual system need to do with borders? Well, it needs to assign each border in the scene to the right side of foreground, such that we can identify the shapes of the objects that are present and recognize the objects. The result is that in perception, the objects that we perceive in visual scenes own their borders. There is a correlate between this perceptual idea of border ownership and neurophysiology. The lab of Rüdiger von der Heydt has discovered neurons in the visual cortex of the primate brain that are selective for this border ownership. So you can imagine that you're recording from a neuron in the brain. It has a receptive field at a certain location in the visual scene. And you can present, for example, a vertical border in that receptive field. But if you zoom out a little bit and you see the entire scene that is present, that border could be part, for example, of a simple square object on the left, but it could also be part of a simple square object on the right. And many neurons in the early areas of the visual cortex, including this V4 neuron that we recorded from, respond differently to these two scenes. So this neuron fires much more spikes to scene 2 than to scene 1, even though the information in its classical receptive field is completely identical. So it is as if the neuron knows that this border in its receptive field is owned by an object on the right side. This property has been initially discovered, as I mentioned, by in the lab of von der Heydt, but confirmed in several other labs, including in our recordings in different areas, V2, V3, V4, where up to 50% of neurons that respond to borders also show this tuning for ownership. An important question, though, is how does the visual system do this? How does it compute the ownership of borders locally in receptive fields? Well, there are converging lines of evidence that implicate the importance of recurrent pathways in the brain. The two major classes of recurrent pathways are cortical-cortical feedback and intra-areal horizontal connections. And there's evidence for both of these involvement in border ownership computation. So as all of you know, the visual system consists of multiple areas in the brain that are organized in a hierarchy. So we start at the bottom with V1, which is closest to the eyes in terms of number of synapses. And then if you rise in the hierarchy, you get to higher and higher areas. So feed-forward connections project from lower areas to higher areas. But there is about the same number of feedback connections that project from higher areas to lower areas. We can differentiate the contribution of these two classes of pathways by looking at laminar organization of signals, because it is known that feed-forward connections arrive in the granular layer of a cortical area, whereas feedback connections avoid the granular layer of a cortical area. And that's what we did in an earlier study, where we found that border ownership signals were first computed in neurons in deep cortical layers and only later in granular layers. And this was done in V4, which is here in the hierarchy. So this means that V4 doesn't simply inherit border ownership signals from V2, where we know there are already a lot of border ownership cells present. But then instead, it seems that deep layer neurons in V4 rely on recurrent inputs to compute border ownership. Other pieces of evidence that point to the role of feedback are summarized here. This is shown, for example, by the pattern of contextual modulation of border ownership neurons of distal stimulus fragments, also of the transfer and persistence of border ownership signals. I will talk about this more in a few slides. And finally, the correlation that was found between border ownership neurons that cover distant parts of the same object. On the other hand, there is also data that supports the role for horizontal connections, namely the dynamical pattern of border ownership signals and the role of context at different distances from the receptive field. In this presentation, I will talk mostly about feedback. So what is the source of feedback? What I told you indicates that feedback seems to be important, but where does the feedback come from? Well, there is this idea that has been floated around since already 20 years that there are hypothetical cells called grouping cells in a higher area that could be the source of feedback, and that endow neurons in a lower area with border ownership tuning. So the grouping cell G2 here in this cartoon, because they're sitting in a higher area, they have larger receptive fields. So their receptive field could cover an entire square object at this location. So this square object would activate cell G2. And because of that activation, the feedback from G2 to lower areas would become active, which projects to certain neurons in that lower area. And that would be an explanation for why this blue neuron here has right-sided preferred border ownership, and the red neuron at this location has left-sided preferred border ownership. So grouping cells are thought to integrate object borders in their classical receptive field and represent a way for the brain to indicate the organization of proto-objects in visual scene that can be flexibly activated. However, the existence of these cells has remained hypothetical. So that's what we try to do in the experiments that I will describe. Is there evidence for the existence of these cells? So where would we look for them? Well, I have mentioned that a lot of the lowest area with a lot of border ownership cells is area V2. So it makes sense to look for grouping cells in V4 because from anatomy we know it's the strongest source of feedback to V2. So we used a property of border ownership to find grouping cells that I will explain first. And this property is the persistence and the transfer of persistent border ownership signals. So instead of showing simple square scenes, as I showed you before, you can make trials a little bit more complex by showing a sequence of scenes where you start with a square object and then you change the display such that you basically remove all the borders of the square except for the one in the receptive field. So you end with just one border that runs through the classical receptive field. And if you started from a square scene with a square on the opposite side of the receptive field, then you will end with an identical ambiguous border. So after 500 milliseconds here, the two displays are completely identical in these two trials. We see in this population activity from V4 cells that there is border ownership signals that start soon after the squares come on, not surprisingly. But after the display switch to the ambiguous border, that border ownership signal remains present for hundreds of milliseconds, even though the displays between the opposite conditions have become identical. And it becomes even more interesting if you add a third step to the sequence, eye movement. So now we start with a square such that the receptive field is not positioned on the border, but on an isoluminant part of the scene. So in that square phase of the trial, we now don't have a border ownership signal because the receptive field doesn't see the border. We go to the ambiguous border again, which is now off the receptive field. And then we move the fixation point such that the ambiguous border lands on the classical receptive field. And interestingly, then you can observe a border ownership signal that appears suddenly de novo, even though these cells never responded to the ownership when the object was still present. At the time it responds, there is no more object and no more stimulus information that would allow you to infer the ownership. So there's this short-term memory for border ownership that persists in the brain, and that can be transferred to other neurons with eye movements. So this was known for border ownership neurons, and we use this property because it results in certain predictions for the properties of grouping cells. Prediction one is that it means that grouping cells in this ambiguous phase of the trial need to show a centripetal preference for ownership. What do I mean with that? Well, if you think about this sequence again, square, ambiguous border, so it's as if the side of ownership remains attached to that ambiguous border for a while. So for border ownership neurons in that ambiguous phase, the border ownership signal persists. And therefore, we expect to see a position invariance if you think about the population of border ownership cells. That is because it doesn't matter for border ownership cells where exactly the border is present in the receptive field. If it's on the left half or on the right half, if the cell prefers leftward ownership, it will always prefer leftward ownership for those different positions. Other cells will prefer rightward ownership, for example, so they will always prefer rightward. So in the population of cells, if you combine data from different cells and borders presented at different positions relative to the receptive field center, you should see get a balanced picture of left and right and up and down preferences. However, the grouping cells should behave differently. They, by definition, integrate borders in their receptive field, so they should always prefer the side towards the receptive field center. So if the border is present in the left half of the receptive field, they should prefer ownership towards the right. Border on the right, they should prefer towards the left. So the result in the population for a similar cartoon population plot here is that most of the arrows should point inward towards the receptive field center. So to test this prediction, we obtained V4 data from macaques while they were fixating visual scenes with laminar probes. This is an example cell where we showed started with a square, and then we, after 500 milliseconds, we changed the display to ambiguous border. And this is the opposite condition where the square is on the opposite side of the shared border. And we analyzed spikes in this gray analysis window here. So you can see that there are much more spikes to the top trial than for the bottom trial. And quantify the difference with this persistent object index, which is simply the difference divided by the sum to the spike counts in that analysis window. So this example pair of trials for one cell is consistent with the grouping cell behavior because more spikes appear whenever that persistent ownership is towards the center of the receptive field. So what does it look like for the population? So this was the prediction from the cartoon that I showed you before, and this is the actual data. So here, every thin gray line is a single ambiguous border at a specific location relative to the receptive field center, and the black arrows indicate the preferred side of ownership in the ambiguous phase. So you can probably see that most of the arrows point to the center of the receptive field. And this results in a bias towards positive values for the persistent object index. Now, an important question, of course, is do we see the same after the saccade happens for these same cells? Because the grouping cell model predicts that the same population of cells access the feedback to border ownership cells in all these different situations, basically. So we added that third step to our experiment, where we start with the squares away from the receptive field, switch to ambiguous border, then move the fixation point so that the ambiguous border lands in the receptive field, and analyze the spikes in that window here. So again, we should see this type of picture if there are grouping cells present in the population that we recorded from. And that is indeed consistent with the data, where again, most of the arrows after the saccade point towards the receptive field center, resulting in positive POI indices for that phase of the trial. Importantly, we don't see this pattern in the same neurons, in the same trials before the saccade occurred. So it's not simply a lingering signal in these cells. And we also don't see this pattern when we do this analysis on a set of neurons that don't obey prediction 1, so that are not quantitative grouping cells. So to conclude, I showed you data that indicates that indeed there are persistent object signals in V4, which is consistent with the presence of grouping cells, and that the same population of cells also shows these persistent object signals after saccades bring ambiguous borders on their receptive field. I didn't have time to talk about a few other arguments that we have that support the existence of grouping cells in V4. Briefly, these are that the latency of these grouping cells are short enough to act as a feedback source to establish border ownership in V2. So we find that the latency is 10 to 30 milliseconds shorter to the latency that has been reported for V2 border ownership signals. These grouping cell signals also survive blank intervals if you show the ambiguous border and then you erase the border, so basically blank screen, and you bring the ambiguous border back. It is known for border ownership signals that the signal can reappear. It will disappear during the blank screen, but then it reappears, indicating that the short-term memory for the ownership was stored elsewhere, presumably at the grouping cell level. And these grouping cells here that we find, they don't lose their signal during that blank interval. And finally, these cells are also less tuned to the luminance of the square compared to border ownership populations. And that's another property that was predicted for these cells. So you can read a little more about these data in the preprint that we have had. And so to summarize, our data indicates that there are persistent optic signals before, and that some of these cells at least may be the source of the feedback that allows feed2 neurons to compute border ownership. And with that, I would like to end my presentation. Thank John Reynolds, my postdoctoral advisor, and my funding sources, and happy to answer questions.
Thank you very much. Very impressive neural data from primates.